Hi. Hi, Grace. How are you? Hi, Chidima. I know I'm on early, not normal. I decided to jump on now and see if I can do a last minute prep for people before the big exam tomorrow. And then I am going to leave you guys to have a good night's rest. So, oh, you're on a school trip. That's cool. Whereabouts? Hi. Hi, guys. If there's anything specific you want me to start covering, then please do let me know. Otherwise, I'll just Oh my God, Chidama, that's amazing. That's so good. Oh, visiting a mine, that's not exciting. You just came from maths. Oh, bless you. How was it? Was it rough? It's a rough paper, right? I'm gonna see if I can actually find it on here. A bit, oh, bless you. Yep, me too. Oh gosh. Guys, it's really hard. It's really hard when you're going from one exam to another. Thank you for your help with bio. Your videos helped. Problem solid and knowledge, especially classification evolution. Enjoyed it. You too, enjoy your trip. Actually, that's an interesting one because classification and evolution, I have a feeling that that could potentially come up. And I was working with a student earlier today and we were going over some questions on that. And I haven't really done much on evolution side of things and classification. So what I thought I could do is let me just bring in a little bit on that today. Um, so one of the bits that I do want to look at with you guys is hi guys, welcome if you are on TikTok, welcome as well. So one of the things they could ask you in tomorrow's paper, obviously I don't know what they're going to ask you. All I can guess is that it is going to be on any of those topic areas, obviously, for module 1, 2, 4, and 6. So therefore, one thing I did like when I saw a picture like this is, do you know how to note where there is a common ancestor? So if they give you a phylogenetic tree, because they haven't done that in a while, do you know how to figure out and how to give some information on that? So, Cara, yes, give me a second. Let me do this first, and then I'll jump over to cell-mediated and humoral response. So if we're looking at this one, so you've been given information about Charles Darwin and a drawing that he sketched in his notebook. And one of the things that they've said is A, B, C, and D. So we can see we've got A down here, we've got B, we've got C, we've got D. And these are all modern day organisms, and they are representing the link that comes from this ancestral organism. So this number 1 down here is an ancestral organism, and modern day organisms A, B, C, and D have all got a relationship between them because they are all linked by branches. But what you should know is when you're doing this, to note where was the last common ancestor. So if I were to put a dot over here, what I would say is that represents the last recent common ancestor. Try that again. Sister of, not A, again, brain engage, B, C, and D. This one down here is the last common ancestor, which is of A, B, C, and D. So you should know about your phylogenetic tree. You should know about your domain, kingdom, phylum, class, order, family, genus, and species. And you should therefore note the fact that A is in a different branch coming off. That means that it diverged away at a much earlier rate, and therefore this one would be in a different taxonomic group. Whereas B, C, and D would be within a more similar taxonomic group because they evolved from a more recent common ancestor. So just be aware, module 4 and evolution can come in lots of different ways. If you get a question like this, they are looking for you to note that B, C, and D have a more common recent ancestor. The evolution of B, C, and D diverges at the same time because they came from that common ancestor here, and A is on a different taxonomic group. Okay, don't be worried. Adam, you're not the only one who's scared about this paper. Paper 2 is always the harder paper of all the papers when it comes to OCR A biology, and that's because it brings in the topic areas that either are really hard to understand, which is the genetic side of things, or where your teaching that you've had of it hasn't linked it together that makes it more difficult. But everybody is pretty much scared of paper 2. Okay, so the humoral and the what am I looking for? Applied passage module 4. If we're looking at the specific immune response, looking at whether it's cell mediated or whether it is humoral, the cell mediated side of things, what you've got is cell mediated immune response is our T lymphocytes are going to encounter a antigen presenting cell. And that antigen presenting cell is going to result in our T helper cells being activated when they encounter an antigen presenting cell. So our T helper cells, when they come across an antigen presenting cell, when they have encountered a antigen, which is a foreign antigen that's been presented on the cell, that will activate the T helper cell. Thank you. To start to produce some interleukins, some chemical signals. And those chemical signals are then going to activate the B cells. So the humoral response is where you've got an antibody-mediated response. So the antibody-mediated response is where, through the process of mitotic division, we will have clonal expansion, and clonal expansion will result in us being able to differentiate to give us plasma cells, and those plasma cells will then produce the antibodies. So we've got to remember that we know there is mitosis, and we know that those cells need to differentiate. And by differentiating into the plasma cells, it's the plasma cell that's going to produce the antibodies. How is the antigen presenting cell produced? Well, the antigen presenting cell can either be a macrophage, which has engulfed and digested, so phagocytosis has occurred. So it can either be phagocytosis by a macrophage, and then it's presented it on the surface, or it can actually be a B cell which has encountered the antigen. It processes that and then presents it on itself to become an antigen presenting cell. And then if it encounters a T helper cell or the interleukins, it gets activated so that it can go through clonal expansion to produce more of itself, and then differentiation to produce the plasma cells, and that then produces the antibodies. So it's important to note the humoral response is this antibody mediated immune response. The cell mediated response is to do with your T cells. Absolutely. As long as you understand the T cells are the ones that are involved in the cell mediated. So, nice. If we've got not only our T helper cells, we also have T killer cells, and those T killer cells will produce perforin. And the perforin is obviously, and it might not be perforin, they may tell you that it's hydrogen peroxide. It may decide in the question to give you something that's slightly different, and that's fine, as long as you are remembering that it is the perforin or the hydrogen peroxide that is going to put pores in the cell. It's going to disrupt the cell, and therefore kill the cell so it can't function. And so therefore what you've got then is also some memory cells. Now, if you think about this T lymphocyte actually being able to go through mitotic division and then to produce the different types of cells shows you that it has to be a multipotent cell because it has the capacity to still go through mitotic division. So that is one of the ways in which we can differentiate between if a cell is able to, just because a textbook doesn't mention it, it doesn't mean it's not that you have to memorize it. It's the important thing is that you understand that the T killer cells are going to produce these chemicals, and it depends which textbook you've got as to what they will explain as being the chemical that's being released. So as long as you know the key killer cells are going to kill, and they're going to kill by producing a chemical, that's what's important. Different textbooks will give you different examples. So the important thing is that you note that it's a chemical that's going to be used to actually kill the cell. So what's the difference between the T clonal expansion and clonal selection? To answer that question, clonal selection is about selecting the right antibody that needs to be produced against the antigen. So selecting the right cell, the right B cell that's going to produce an antibody against the antigen. So the antigen and the antibody are going to have to be able to be complementary so that when they come together, what that's going to do is that's going to therefore have that bonding because of we've got this complementary shape, the antibody antigen complex. So when you talked about an enzyme substrate complex, that was because they had complementary shapes that they bonded together. So when we're looking at the antigen antibody, when they come together, we have an antigen antibody complex. They are complementary. So when we do clonal selection, we need to select the correct B cell, which has the antibody, which is complementary to the antigen. Clonal expansion is about expanding, making more cells, more copies, clones of the original one, because that's the one where it's going to produce the antibody that we want. How do you go about revising evolution classification? Go check out my videos on it. But basically, it will be very specific to the example that they decide to give you in the exam. Yeah, phylogeny is basically based on classification, and classification started off with anatomical structures. Then we went to embryology, the study of embryos, looking at how they developed, our Hox genes, for example, and then looking at the DNA sequencing. So when we talk about phylogeny, we're talking about the evolutionary relationships, which is based off classification. And how we do that classification is based off all these different structures. What do perforins do? So the perforin is the chemical that's going to come in and burst the cell apart. So basically, if you've got a cell which is infected, let's say there's a virus inside, and that virus is going to be using the organelles inside to make more copies of itself, using the enzymes to make more copies of itself. That's therefore going to have a way in which it will then burst out of the cell, but taking along with it some of the membrane. Well, the T killer cell comes in and bursts basically like a big balloon that's got something fired at it. It will burst open. Those organelles and all those different structures are now free floating. It cannot use them and utilize them in the right way. So basically, you're destroying your cells. And I'm very lucky, although I sound rough and although I sound like my throat should be hurting, I don't actually have any of my cells being destroyed at the moment. I don't have the scratchy throat. I don't have any of that. Right now, what's happened is I've overproduced the mucus. My goblet cells have produced too much mucus, and it is therefore coating my larynx. And what's going to happen is that coating then makes the vibrations work slightly different. So I always end up with laryngitis, something that always happens to me. But when I start to be able to clear the mucus that's in my lungs, that's going to therefore free up and allow the vibrations, and my voice will go back to its original sound. But right now, it's not obviously doing that because right now it is sounding very coarse. I always have to bring the link. I always bring the links back to real life situations because that's what it's about. Um, does the hydrogen peroxide break cell membranes? Yeah, absolutely. It's going to disrupt the cell membrane. So basically, you are breaking apart. The hydrogen peroxide will obviously get in, it will disrupt the membrane, it will therefore cause the cell to not be able to function. Yes, give me a second. Wait, is thought it was the interleukins which activated the B cells. So the activation of the B cells is from the interleukins, but you've also got to remember that the antigen could have actually been encountered by the B lymphocyte. That therefore means that the B lymphocyte, if it's got the right antibody, it itself can become an antigen presenting cell. Because you've got to remember, you don't just want to have one cell. If you've got memory cells there, you're going to therefore want to activate them as well. So the interleukins are going to activate the B lymphocytes to go through their clonal expansion. But you also remember interleukins are going to activate our signal for your phagocytes to come to the area. And so the phagocytes coming to the area will therefore help with the engulf and digesting. What could they ask about herd immunity and epidemic stuff? Well, they could give you examples about vaccination programs and talk about the fact that if somebody is living in an area where there is a big outbreak, how could we reduce the spread of it even more? So herd immunity is an interesting one because the herd immunity side of things is where the COVID vaccine came in. COVID vaccine was obviously given so that the majority of the people who are vaccinated have an immune response against the pathogen when they encounter it. Therefore, if there's enough of a protection within the community, there is therefore going to be less chance of it being spread. So the herd immunity side of things is that if enough people get immune against whatever the communicable disease is, the pathogen, what that therefore would do is reduce the spread of it going throughout the locations. So when we talk about epidemics, we're talking about it spreading to other countries. And so therefore, by doing the isolation, by keeping people away, by having the wearing the mask, etc., that therefore reduced the number of cases. But now we're back out into freely roaming around. We've now got different versions because of mutations. Mutations therefore means that you have variations in the pathogen. So that's why vaccinations each year have to be done for the flu vaccine. So a phagocyte can become an APC after it's carried out phagocytosis, but phagocytes can also carry out phagocytosis. So you've got to remember it's about the chicken and the egg. It's about which one comes first. How does that link to bioinformatics and computational biology? Well, they could link bioinformatics and computational biology into anything. Do I think that's coming up as a huge question? No. I do feel like the question could say about the fact of bioinformatics was used to compare the DNA sequencing from lots of different organisms from different locations. So if we were looking at epidemiology and we were looking at the spread of a disease, what we could say in a case is that actually what's happening is if we have a variant and we want to see if that variant in the UK is the same as the variant in Portugal and the same as the variant in Tunisia, what would we want to do? Well, we'd want to sequence the variants that are located in those areas, and then we'd want to compare their DNA sequencing. So this is where they could say about how do they know that it's spread? How do they know that it's gone from one place and it's actually spreading elsewhere? They do that through DNA sequencing. By looking at the DNA, they can then compare to see, is it the same variant or is it different variants? Comparing it to other ones. What else? I haven't really made any predictions. I'm just literally going off the questions that are on here. Um, do you need to know about like A, Y, and Z for like operand? You need to know about the fact that there's structural genes. Do you need to know the specific examples? I feel like they will give you the information if they want to ask you questions on it. I don't feel like it will be specifically you have to know the like Y, A, and Z. Um, sorry, give me 1 second. Predictions. Cytokinin, yes, are a type of interleukin. Absolutely right. I want to, somebody did ask me something that I wanted to pick up on. Where are you? What did you ask me? You asked me something up here, which wasn't related to this, but was important. Do, 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 do. Phylogeny is the evolution, right? Let me put a bit about phylogeny in here because people are worried and I haven't done that much on it. So let me do that now because I do feel like they will bring in a bit of phylogeny and I do feel like they will bring in a bit about how that links with the evolution of organisms potentially. So phylogeny is basically the name given to an evolutionary relationship. So when we look at relationships between organisms, when we're looking at, for example, us and chimpanzees, we're looking at how we may have evolved from a common ancestor. And so to be able to do that, it is based around the basis of classification. So classification is going to help us, the same grouping, how these groups are being put together because they share these common ancestors. Now, they don't ask you about understanding what a clade is. So this is where they give you a little bit of information, but they don't give you or expect you to memorize enough of it to make it make sense. What's important for OCRA is that when we use a phylogenetic tree, we are showing this relationship, this evolution relationship, because we're saying if they're connected together, if they're branched together, they have therefore come from a common ancestor. So these branches that you see coming off each time, they are showing you different species that evolved from the last known common ancestor. So all the way down at the bottom here, we have got a common ancestor to all of the organisms in the phylogenetic tree. When we look at any place where there has been a branch that comes off, that is the last known common ancestor of all of these descendants. So that was the last known common ancestor of all of those descendants. So therefore, something has happened which has resulted in new species. So therefore, what has happened? Probably mutations, which create variation. Then we have selective pressure. So this is where evolution needs to link back to natural selection and needs to link back to how we can tell how organisms have evolved. So when you look at, for example, how we classify organisms, we start, oh, I got stuck. Sorry, guys, I missed a whole load of questions there. But how we look at classification is we start with anatomical evidence, looking at the structure and function of different body parts. So the pentadactyl limb, so I have 5 digits here. I have the pentadactyl limb, and therefore I have my humerus, I have my radius and my ulnar. So my radius is connected to my thumb because my thumb can make an R. So I have my radius bone, which connects here at the front. Then I have my ulna, and then I have my carpals, my metacarpals, and my phalanges. So I have my 5 digits. I have a pentadactyl limb. Here, I have my pentadactyl limbs, which are my legs that go down to my metatarsals, my tarsals, and my phalanges. So when we look at the pentadactyl limb, we are looking at homologous structures. They are homologous, homo, same. So we have the same structure. So we've got the humerus, we have the ulna and the radius. So if we have organisms who all have the pentadactyl limb, they all have these same bone structures, they therefore have these similarity in the shape, in these different types of organisms, despite being used in different ways. So we've got a bat that's going to fly, we've got a dolphin that's going to swim, we've got an anteater that's going to tear, we've got the monkey that's grasping, we've got the pig that's walking. So these homologous structures, the more similar those homologous structures are between the two species, when we look at the anatomical evidence, the more closely related they are. So when we talk about homologous, I'm going to cover that in a minute, Grace. So when we talk about homologous, homo same, homologous structures, similar in shape. So therefore we have the humerus, the radius, the ulna, the carpals, the metacarpals, and the phalanges. The more similar they are in structure, the more closely related. That's how we can classify them based on anatomical structure. And that is showing you adaptive radiation. Yes, there's changes because they use them in different ways. So the bat's second, third, and fourth digits are much longer than the first digit. And that's where the membrane attaches, which gives them the ability to wing, to be able to fly, and gives them their wings. So therefore, excuse me, homologous structures are a way in which we are saying they are similar because of common ancestry. So therefore, we are showing divergent evolution. If we are looking at these organisms, and you should hopefully know that all of these will be classified as being mammals, they have evolved from a common ancestor because they have very specific structures like that. So when we look then at, oh, no, I didn't put it in. Hold on a second. This is where I've got to move stuff across from my other ones. So you asked about analogous structures. So if I'm looking at analogous structures then, here we have a bat, which has the wings for flying. So you can see the structures here. You can see the bone, the digits coming out, and then the membranes that are attached. And then we've got the bird, which has a completely different structure. However, they are very similar, but also different. So this is where you have analogous characteristics. So they share common ancestry because we'll go all the way back. However, they have actually evolved not the same, not from a divergent from a common ancestor. What's happened is they have similar structures to provide similar functions, but they evolved these independently. And they evolved these due to having similar niches. So when we look at the wings of the bat and the wings of the bird, these are analogous structures. But if we were looking at the pentadactyl limb, those were homologous structures. So it's important you're looking at whether you're talking about analogous characteristic, whether you're talking about a homologous structure as to what you want to talk about when you move forward in that classification. So these are convergent because they were because of similar niches, similar habitats, and similar lifestyles. Okay. Good. Yeah, selective pressures are important. When would I need to talk about convergent and divergent? So convergent and divergent evolution would be looking at where you have, so let me just go back to the, no, analogous would have the, when you say analogous have different functions, well, if we're looking at the bat and the bird, their function is for flying, right? So the function is to fly. So therefore they have a similar function, but you look at the different structures. So we've got the membranes here, which are attaching the digits, whereas here you've got your feathers, which are attached to different structures. So homologous for divergent, analogous for convergent, absolutely. So if we go back to our phylogenetic tree, and if I look at where the, see if I can identify, hold on, actually, see if I can bring this one in here. Turn it on its side. slightly. Um, no, that one doesn't have what I want. Hold on, give me 1 second. I'm going to bring a bit more information that I'm hoping will help. Give me 1 second, guys. I'm just going to add in a few slides on previous things that I've done in the past. Right, okay. So let me move past this one. It wasn't well. Okay. So we were talking before about the phylogenetic tree. So you can see the descendants 1, 2, 3, 4 here. You have your ancestor, your past, and to the recent. You can see wherever there is a speciation event. So what could happen is they could show you there's been some form of speciation event that's occurred at that point there. So you've got the ancestral lineage here where there are changes. Just to be aware, there are still mutations occurring here that are occurring all the way up here that are changing the organism, but we don't have a speciation event until we get to that point there. So that's the first important bit. So then when we look at the homologous structures, so in this case, in this example, I've got 4 limbs have evolved at this point here. So these are called tetrapods. So therefore we know that the 4-limbed individuals all had to have evolved from a common ancestor. There've been lots of speciation events occurring that have resulted in changes, but they all had to evolve from a common ancestor. That common ancestor, it actually was the one which evolved having 4 limbs. So that's where we'd be talking about homologous structures, similar because of that common ancestry. And obviously, when we look at the analogous structures, as I said before, convergent, similar structures to provide similar functions, but evolved independently of each other. So you can see here, 4 limbs evolved back at this point here. However, you have the bat over here and the bird over here. So they both have the similar structures. They both have similar functions, but they evolved independently of each other. Technically, it isn't always gradual, but in your case, yes, we would talk about it as being gradual for your exam. Right. Common ancestry, different function, analogous, similar function. Yes. So allopatric speciation due to geographical isolation. Yes, some form of geographical isolation is allopatric. Sympatric is some form of reproductive. So you're in the same environment. There's nothing which is separating you, but it is actually causing that. I feel like DNA and DNA replications coming through and how they ask the question could be go in any direction that they want. Okay, I'm just making sure I haven't missed anything. How long am I going to go live from? I'm probably going to do, I could probably do about another hour, I would say, to help as much as I can. And then I'm going to close off and I'm going to enjoy the rest of my evening, as you should. Have a rest, have a break, but also make sure that you are sleeping well, eating well, and also make sure that you are not stressing too much about what could come up, because I think this is the one paper where I don't feel confident with predicting it. The only thing I would say I feel confident predicting on the side of things, I feel like gene regulation is going to come up, and I feel like they could ask it in such a way that you don't realize what they're asking, but I don't know. How do you, so, okay, let me just finish this one off. Anytime you talk about speciation, okay, I go up here. So anytime you want to talk about speciation, it's really important you always think about some key things. So speciation has to be some form of isolation. Now, what is that isolation? What has caused the isolation? So we don't know. 1 second. Oh, we don't know what the isolation is yet. I haven't got the question. I don't know what the isolation is, but something has isolated the organisms. Whether that's geographical or whether that is reproductive, there is some form of isolation that has occurred. Now, we then want to jump back to natural selection. So natural selection is obviously where we're going to add in some keywords. So natural selection is mutation, variation, selective pressure. Then you have survival, you have reproduction, you therefore have alleles passed on, and therefore the frequency of the alleles will change. So natural selection always have those words: mutation, variation, selective pressure, survival, reproduction, alleles passed on. The frequency of the alleles will change over time. And then when we talk about evolution, evolution is the accumulation of these changes over time. The accumulation of these inheritable characteristics over time. Wow, my writing is atrocious. So speciation, there is isolation that happens. Then we're going to have random spontaneous mutations that occur because they happen in DNA replication. There's going to be variety, variation within the population. The environment will give some form of selective pressure. Now, that selective pressure could result in, so I'm going to bring in a little bit here on selective pressure because I do feel patterns of natural selection are coming up. So I do want to cover this for you guys. I don't feel like I've done that enough. 1 second. Let me just bring in the slide I want to talk about. Where are you? Where are you? Where are you? Think through, what am I doing? Patterns of inheritance. Inheritance. And I want selective pressures. So, oh, I went too far. I will come back to that slide. So natural selection, increasing the frequency, keywords, mutation, variation, selective pressure, survival, reproduction, alleles pass on. The frequency is going to change. So therefore, when we talk about selection pressures, we're talking about environmental factors that are going to act selectively on phenotypes. And they're going to act selectively on the phenotypes, resulting in natural selection. So there are 3 patterns that you should know about. So it really depends, Tony, with regards to sympatric speciation, it just depends what is going to be the environmental factors. So if there is, so if we're looking at reproductive isolation, if they're within the same environment, there's no geographical isolation, what could happen is they could, let's say, flower at different times. So it could be a mutation that results in them actually flowering because they are detecting the light changes at a different point. And so therefore they are going to flower at a different point, which results in them not being reproductively available at the same time as the other plant species. So in that case, you are reproductively isolated within the same environment. It could be that the egg and the sperm, or the egg, ovum, and the pollen are not able to actually fuse. So therefore, you've got a problem that the pollen tube isn't being produced in the right way. There's so many different ways that they can go with that. It just depends. So sympatric is about them being in the same environment, so they are reproductively isolated. It could be behavioral. So it could be, for example, that they don't have the right behavior. It could be the bird song. They have a genetic template that they have where they know the template of the song. And what they then do is they then listen and learn and they mimic. But if they are, let's say, surrounded by no adult birds that sing the song that they are going to mimic, they can therefore result in a different song. That different song results in them not being able to mate with the same one. So it's that whole thing of, it really depends on the example that they give you as to what they may actually ask. But there are three patterns you need to know about when it comes to selective pressure. So they're going to be either directional. So the peppered moth is a really nice example, because Industrial Revolution, what happened? Industrial Revolution kicked up a whole load of particulates. The dark trees now, the dark peppered moth is going to be camouflaged. Camouflage, what are we talking about there? We're talking about a anatomical adaptation, which is module 4, anatomical adaptation that therefore results in them not being predated. They survive because they're camouflaged. The light pepper moths are going to die out. Because they're dying out, they're not able to reproduce as much. And so therefore, the alleles that are being passed on is going to result in more of the population having the dark color than having the light color. Well, we've got better at what we've done. We've cleaned up our air. Because we've cleaned up our air, what we're now finding is there are less particulates, less soot, less coating of the trees. Now we've got more of a directional movement towards the light peppered moth. So the selection pressure could be directional in one way or another. So maybe going to the dark side in the case of the peppered moth, or maybe going to the light side in the case of the cleaning up of the air. With regards to, excuse me, stabilizing selection. So the one that we tend to talk about is about birth weights. So stabilizing selection is that we're selecting for the average. Now remember, this is showing you a normal distribution curve here. So you know that your mean is going to be whatever that mean is calculated. And then what you have is you have plus or minus 1 standard deviation. Now that is normally 60, oh, I'm going to say 68%, 65 or 68%. I'm doing this off the top of my head, so I may be wrong on this one. Correct me, guys, if you remember off the top of your head. I want to say it's 68% is 1 standard deviation. 2 standard deviations is 98%. So therefore, you will get a normal distribution, which is what my graphs are showing you. The normal distribution is about whether that distribution is changing. So where's the mean going to? In this case, with it being directional, it was moving in that direction. Oh, good. Thank you, Lisa, that I was right with 68. Oh, maybe it's the 98% that's wrong. Is it 95.8%? Anyway, I haven't taught that in a while. I used to have to know it for the IB, and it's out of my head at the moment. Anyway, back to this stabilizing. When we talk about stabilizing selection, we're saying that we are selecting for the average. So therefore, what this is the stuff that I think is going to come up. Ah, there we go. Thank you. I knew there was a 5 there somewhere. Thank you, Amna. 95%. That would make more sense. For 2 standard deviations. So if we look at birth weights, okay, so birth weights, the average that is selected for is £7. So £7 is the average birth weight of the majority of babies that are born. Okay. Now it doesn't mean you don't get birth weights which are lower and that you don't get birth weights which are higher. It's just that the average is the middle. Now it says here, the stabilizing selection, we are selecting against the higher ones. Now, what does that even mean? If you have a lower birth weight, then it will mean that the embryo and the fetus has therefore maybe not developed enough and therefore is more likely to survive, to not survive, sorry, is more likely to unfortunately have complications and to die. So you can have a few of them, but you don't have a lot of them. Go too high, and there's a problem with the birthing process, and they're more likely to die through the birthing process. So therefore, the environmental factors that are acting selectively, £7 is the average birth weight that is more selected for, because more are going to survive because they are developed enough and they also have the passage in the right way that helps them to get out the birthing canal without any complications. So in that case, we are selecting for the average, stabilizing, selecting for the average, and selecting against the extremes. Now, if we were to look at disruptive is where what you end up with extreme phenotypes resulting. So we are ending up with the extremes and we are selecting against the middle. Now, in this case, I've said finches, but I'm going to talk about a different example because I really like this example. And I feel like this one tells it more than the finches, but for some reason, Darwin and the finches are always the questions they go to in the exam, but that doesn't mean they're going to do it this time. But I love the one to do with salmon. So the coho salmon, there are different types. You can have your jacks, which are really small, or you can have your hook noses, which are really large and have these hook nose at the front. So that's what they're called, the hook noses, and you've got the jacks. Now the jacks are really small, but the jacks actually get to that reproductive maturity at a faster rate than the hook noses. So the hook noses take longer to get to reproductive maturity, but they're much bigger, so they're the better competitor and they fight out any mediums. So the medium sized fish are actually selected against. The small ones are selected for because they're really cunning. What they do, because they get to reproductive maturity at a faster rate, they sneak in, they therefore release their sperm. So they sneak and hide away from the hook noses, release the sperm, and off they go. The hook noses outcompete the mediums, and so therefore we end up with the two extremes with the population. So you have the smaller ones, but you also have the larger ones. It's not that you do not have the ones in the middle, it's that they're selected against, so the frequency for them is smaller. So all three are in this spec. So you should know all three. They are in the specification: directional, stabilizing, and disruptive. See, now you said that, I'm going to double check because now I'm like, hold on a minute. And I always do say, always go and double check against the specification to make sure that you are covering all the different pits. But I would double check because now you've made me question it. think. Let me just double check. Um, and it is always good, by the way, guys, to actually double check, excuse me, with regards to that. Because actually, I'm just going to double check on the. This could be a bit where I've actually missed. Nope, you're absolutely right. It's not on with disruptive. It is actually just stabilizing and directional. You're absolutely right. Doesn't mean they won't ask the question though. So, but Zed, thank you. It is pretty simple, but in a sense, they will potentially give you stuff because I have seen it in a question before. So that's why I'm surprised. I feel like it's in a question, even though it's not in the spec. I do feel like it's in a question. Otherwise, I wouldn't have put it into the resources. But I may have done that. I'm going to just double check. Wow, it is hot today. Patterns. No, so the only question I can find now, because I'm trying to find it on the spot, is the one about stabilizing. But I swear they've given one before. So hold on, let me show you. It's actually a really nice question as well. And it was about the parts to do with the finches. They love a good finch one. So this one is the type of question. So this one came up in 2021's paper 2. It was a 6 marker. The one I'm going to show you. 1 second. Let me go back. So this was a 6 marker and it was about stabilizing. And so therefore, they're looking for you to use the data. So the results suggested that stabilizing selection was occurring. So therefore, the minute you see it say stabilizing selection, automatically you need to be saying, right, the mean selected for, and then you want to be looking at extremes against. And then you want to relate it to the question. So with reference to figure 20.2, explain the effects of stabilizing selection on this. So therefore, what you're looking at when you see a question like this is you're looking at the fact, well, what causes this change? Well, remember, you need to have genetic variation. So where does genetic variation come in? So I've talked about mutations. The genetic variation is not just about mutations. We're talking about variation. We have to bring in the fact that it could be to do with sexual reproduction. It could be to do with the fact that which sperm and which egg were actually fused together, which of the two gametes. So if you're not sure what to describe as the sex cells, use the term gametes fusing. And the reason I say that is because gametes can refer to whether we're talking about pollen in a plant or whether we're talking about sperm in animals. So it's important that you use the words that you feel comfortable with. And if you're not sure, then don't use the word, use the generic term. So a gamete is a sex cell. So we've got those gametes that are being fused, but then where did the gametes come from? Meiosis. So then you need to think about, well, okay, what is meiosis then? Meiosis, that can result with independent assortment. Variation can happen because of crossing over. If they are linked genes. And obviously the random fertilization, which links to that sexual reproduction, random fertilization. So these are all going to increase genetic variation, but then we need to talk about, well, what do we have? Well, we know because of Darwin's theory of natural selection that there is an overproduction of offspring. So therefore, there is going to be an overproduction of offspring. And because there's an overproduction of offspring, oh, hold on, have I not changed my slide and you guys have not seen this? Oh, no, you have. It's just lagging on YouTube. So when you look at the natural selection and you're looking at the fact that you have got the what was I saying there? Overproduction of the offspring. You have the fact that those extreme beak depths are less likely to survive. So you can see that in the diagram, they're less likely to survive. How do we know? Because of the fact that you can actually see that when the beak depth is too high, there's very few individuals found with that beak depth. And when we go the opposite way, there's very few found there. So we want to talk about the change in the frequency so that you can see that that is, so up here, there's very little, down here, there's very little. And because of that, that's therefore telling us that the extremes are not being selected for. Instead, it's the middle ones. But we have some random numbers out there. So do remember that you also have the other part of this where you're looking at whether or not there are some that don't follow the expected trend. But overall, this graph is showing stabilizing selection because the mean is selected for, the extremes are selected against, and therefore we're linking it to the information in the graph and we're using that to help us. Yes. So we know that there's always overproduction of species, and then what's going to happen is competition comes in, and that's where you've got the selective pressure. Is the selective pressure to do with the is it to do with the amount of food that's available? Is it to do with the resources available? Synthetic biology did have a 6 marker link to it, but you do have to still learn it. You can't assume that just because it was a 6 marker last year, it won't come up. It will come up, but the way it comes up will be slightly different. It won't be a 6 marker, but it will still come up. Yeah, you do need to know it. It's about personalized medicine because basically, how could they bring in synthetic biology? Well, why would they do PCR? Why would they do DNA sequencing? Well, what they could do is they could make medicine that's personalized for someone's DNA. So they would obviously sequence the genome and then ensure that the medicine that they are producing is specific for that, whatever that is that's the problem. So if it were, for example, they are looking for a specific protein that is causing problems, then they could have a chemical that's going to bind because it's going to be a complementary shape to that protein that is, let's say, a receptor on a cell. It would be more linking it to, for example, a specific disorder they wanted to look at, and then looking at the fact that you're making the medicine, the chemicals that are going to be able to be specific for that gene. No specific, absolutely no specifics are required. Can I just remind everybody who, if you're on TikTok, if you're on YouTube, can I remind you this? I've been saying it to my students all day today. The mark scheme that you are about to sit the exam for is not finished. And what I mean by that is, yes, there is a mark scheme that is ready, waiting to go to mark your exam papers. But what will happen is the senior examiners, after you have sat the exam, will sit down with the papers, with the mark scheme, and they will then look at what you write as your answers. And they will see some common threads coming through from you. Now, there's 20 odd thousand of you potentially that are going to sit this exam. So what they will then do is they will come together as senior examiners and they will then agree what they will allow, what they won't allow. So therefore, they will modify that mark scheme for the exam that you are sitting that year. So the years before, those past papers that you have used, those mark schemes that you have used, have been specific to the exam paper and the students who sat it that year. Okay? So this is one thing I would say to you is the mark schemes will be adapted. They've got the ideas that they want, they've got the key science that they're expecting, but you'll see allow this, do not allow that, allow this, do not allow that. And that's because they have looked at what a lot of you have written and gone, actually, yeah, do you know what? We didn't have that as the set mark, but we will allow it. Or, do you know what? We won't allow that because it doesn't make sense. It's not true. It's not the correct science. The other thing I will say to you as well, when you're in that exam, go with your gut. Those of you who are overthinkers, trust your gut, trust your initial answers because normally your gut is right. The next thing I will say to you, if you don't have a clue, if you are looking at that paper and that question and you're literally going, I do not have a clue what this question is on about, write anything you think that you could potentially get marks for that is slightly relevant to that question. Write it down because if you do not know, but you write something down, you might have one marking point worth in there that the examiner can give you. And you were like, huh, I was trying to be funny by writing that. I promise you, write it down because you never know. Your guess might actually be from the back of your brain. Some knowledge you didn't know was actually there, and it is true to the question. Do not leave any questions blank. Please do not. Even if you think that you don't have a clue what to write, just start writing a load of drivel that's biology that you learn, whatever it may be. Can even be GCSE biology, just write something because I promise you, an examiner would much rather you do that. They're not sitting there going, ha ha ha, look at this ridiculous child. How could they have written that? How dare they? They're not going to do that. Okay. What they're going to do is they're trying to find a way in which they can help you. So they are looking for some marks, but if you don't give them anything, there's nothing they can do. Neurocommunication cannot come in. It's not paper one. It'll be paper three if it's coming back in again. Neurocommunication was technically in paper one. You just didn't see it the way you wanted it. Neurocommunication was your cardiac cycle and your electrocardiogram. That is looking at the neural pathway. So it was there. You just didn't see it the way you wanted it to see it. Okay, so this is an interesting one. This is what I would say. So when I'm saying write anything, That is if you have no clue. If you write something correct and then there is something incorrect in your answer, they are going to penalize you and you will lose a marking point for that. So I'm only saying to write something down if you are unsure, you do not know how to answer the question, then write something down. But if you are taking the time and going through, then you should be able to find where they're actually trying to get those marks or those answers from. My daughter's banging at the window at me because I'm normally inside. If I was allowed to talk about hyperpolarization in section, no, hyperpolarization you wouldn't want to talk about. Depolarization and repolarization is what you'd want to talk about. Remember, it could be that it's an ignore. So there, it may be that it's an ignore point, that it's not a do not allow. If it's ignore, we ignore that you've written it. That's the other thing to know. Do not allow is incorrect science. Ignore is different. Ignore is that we will ignore that you've written that and we're looking elsewhere. But if you've written incorrect science, then that is going to be disallowed. No, Tony, I'm not allowed. Because they have not been released, I shouldn't be going through them and I shouldn't allow that. And actually, it's not fair because some students are still going to be sitting their mocks if they're in year 12. And so therefore, I'm putting them at a disadvantage by going through those papers. Any advice on, yeah, let me just do a quick one. How are teachers allowed to see them? Because teachers are allowed to see them so that they can look at where misconceptions have happened in the questions in the previous years so that they can then guide their teaching to improve their teaching to help students be able to get better at it. So it's about helping the student, the teacher, so they can help the students basically. Right, evaluate is absolutely, you are right. We want a for, we want an against. Does support, does not support. So anytime you're looking at evaluate, it is really important that you are talking about where there is evidence that is supporting whatever they're claiming it to do and where there is evidence against, so not supporting whatever it is that they are claiming to do. So the one thing I would say to you is any evaluate question, always do things like this. Look at what's their independent variable, what's their dependent variable, what are their control variables if it's an investigation. If it is sampling, what type of sampling have they done? Is there any type of bias that could invalidate the data they've collected? So independent variable, dependent variable, control variables, have they given you information? What are the intervals? So if they give you, for example, someone mentioned about epidemiology. Oh, I can't even say that word right now. This is where my brain is shutting down because we're getting to, how long have I been going for now? I don't even know. 68 minutes, over an hour. So if we think about the epidemic that went on, so if we're talking about epidemics and we're talking about how the spread of something has occurred, when did they look at it? What years did they do the sampling? How many years have they got between? Is there 4 years between the sampling data or is there 1 year between? Is there 6 months between? So it really depends on what they're giving you. Look at that data. Do you think those intervals are close enough to give you a good set of data? A valid set of data that you can then draw a conclusion from? Have they given you a graph? And if they've given you a graph, for example, if they've given you a graph, have they actually given you your error bars? Are the error bars small? So is the standard deviation small? Or is the standard deviation large? So is there a lot of variability in the data that they've provided you with, whatever data they have? Have they actually taken data at the same points and asked you to compare? So for example, if I do this one, I've taken 3 sample points, point 1, point 2, point 3, whereas on the previous data, I took 5 sample points. So therefore, that's 5 sample points with smaller intervals is going to give me a more representative set of data than if I take 3, for example. What else would I say? Evaluation. Have they done a stats test? If they haven't mentioned, you should do a stats test so you can say whether there is a significant difference or not. And what else would I say? If they make a claim, have they given you all the information? And if they haven't given you all the information, what information do you need to be able to do that data? Right, give me 1 second, because someone asked earlier about histograms, and I have a feeling they may ask you about a histogram. You're going to draw a graph because you haven't drawn a graph yet. So I feel like you're definitely going to draw a graph in your exam. And so therefore, I would say, let me just very nice. Okay, so if you were looking at the, let me just make this histogram, here we go. So if you're looking at a histogram, one of the things that I would suggest that you do is note that histogram is like a bar chart, but it's for continuous data. So therefore, it's really important that you note that because it's for continuous data, it therefore means that you're going to group things into classes. So you've got those intervals. So whatever those intervals might be, and therefore you're going to end up with a frequency density. And that's what you're going to be plotting out, is that frequency density. So then you may be asked to process the data. So if I'm asked 3 out of 5 to get that frequency data, that would therefore be 0.6. And then you would then plot it out. Now it's important to note that the histogram has a title. The y-axis is your dependent variable, which is my frequency density. My x-axis, go away, is going to be my leaf length in centimeters. And then what I want to do is I want to ensure that my bars have no gaps between them. And also, please, please, please do not get caught out on this. Your width of your gaps of your bars should match your classes. So here I've got a really small one because I'm going 0 to 5, but this one is larger because I'm going 5 to 15. This one is even larger because I'm going 40 to 60. So do make sure that when you do a histogram, that your width of your bars actually match the class width. No, it depends whatever they give you. So it depends whatever they've given you in the question. So let me show you one which let me just quickly find it. I'll go back to this one. Let me show you one which was in a paper. I want to say it was the 20. Oh, 2022 paper, which was actually on fisheries, and I don't feel like they've done a lot on fisheries. Sustainability. Oh, hold on. Oh, I can delete that word. Um, oh, this is really annoying. Now I'm trying to find it. I can't find it. I'm gonna go a different way in a second. Oh, maybe it's 23 then. Not 23. What did I say? Oh, 2020. What am I doing? My brain's not functioning. My brain is literally switching down. Um, this is the other thing I was saying to you as well, guys. Right now, you're there we go, found it. Um, right now, you're trying to do lots of revision, and your brain will only be able to cope with so much. And if it gets overloaded, it will then start to shut down. You won't be as effective. So do make sure you have an early night tonight, that you're not just working your way through and doing lots and lots and lots, because it's not going to help you. Right, I've just lost it. Where are we? So this was the question. It was in 2022, and they then wanted the students to look at the Atlantic cod and look at the body length and the frequency of it. And so therefore, they are going to ask you in that exam, they ask them to do a histogram. And so if you understand that you could be asked in the graph, as they in the paper, even as they did, you could be asked to draw a histogram of that data. It is important that you know that and that you're able to do that. So when there's a question about, for example, sustainability and about fishes, they're looking for you to do this information. And then they talked about in the question, it was a really lovely question, actually. It was looking at variation with fish, because it's a paper too, they can look at how different variation can occur. And so therefore, in this case, they wanted you to then take your knowledge about the fish, and then there was like one question, hold on, let me find it, about commercial fishing, which was a 3 marker. And then there was one question later on, which was a 1 marker. So what they're doing is they're assessing that part of the course. They're assessing that part of the course based around lots of different areas. Uh, So southern blotting is a part where they can still ask questions about it. But you need to be aware of it because it was a name they were expecting you to say in the exam. So microarrays are actually, that's more AQA or other exam boards. That's not OCR A. They are not looking for that. 2022. Yeah, it's an interesting one, right? Um, I do feel like I would agree a lot of nucleotides are coming up. Yes, I would agree with you on that one. I feel like they're going to look at nucleic acids because they haven't done very much on that one. And that one covers DNA replication as well as what do you call it? Replication and protein synthesis. And that links very nicely to mutations, which is module 6.1.1. But remember, the one thing I would say to you, they're never going to use those exact same questions again. The topic areas could be similar, but they won't use the same question. So they won't be exactly the same. So don't all of you go, oh, I'm now going to go and do 2022, because that's not going to help you. Can I go over the histogram one? Yeah, of course. So with regards to this one, what I'm looking for is you need to know they're really sneaky in what they've done. So here you can see 100 to 200, 200 to 300, 300 to 400, 400 to 500, 500 to 600. This one is 600 to 800. So this one is obviously got more and therefore should have a larger bar. So the width of this one should be larger. So then you need to think about, okay, if I'm going to draw this, how am I going to draw this? What's my scales going to be on this one? So that's what they're looking for. They're looking for, do you actually know the scale of how to draw a histogram? So it just depends, whatever they give you in the exam, they're never going to give you this again. But what they're looking for you to note is on your, where's it gone? That one, that's what I want. On the part of what they're looking for. What they want is, have you got this title on your X axis? So this down here should be there. Have you got this, my Y, on this? So that, and do not skimp with what they've written. Do not abbreviate. So body length in millimeters goes there. We then have our frequency, which is going to go here. So you can do frequency or you could do frequency density here, depending on if that's what you want to do. It's your choice whether you just plot frequency or you plot frequency density. Because in the paper, let me just confirm on this one. Does it say, just says plot the results. So you could do either or. So then what you want to do is you then either plot the frequency or you're plotting the frequency density. And then what you want to do is you then want to ensure that you are using more than 50% of the graph paper. So your histogram bars should be big enough to cover at least that, okay? So don't make it too small, but don't make it too large that it's not a good scale. So we're looking for a linear scale. So if I'm starting this and I want to go from the 100 and make my way up. So I would then want to plot using that Just double checking what the extra marks were. Yeah, twice the size. So then I would do, it's really hard to do because I made it so small. So going along, count how many boxes you want. 1, 2, 3, 4. So I've got to make it bigger. So if I want to do 1, 2, 3, 4, 6, 7. If I want to have a width here, which is 100, so therefore I'm going from 100 to 200, then I need to make sure that I can fit all the way up to 800 in my scales. So okay, so if I'm doing 10, so I could potentially go small again. So yeah, so I could actually start it at 0, 100, 200, 300, 400, 500, 600, 700, 800, doing 10 squares going along. And then what I need to think about is depending on whether I've used frequency or frequency density, going up, what's my highest? Well, if I'm going up to my frequency, my highest number is 130. So I need to make sure that I can do my scale up to 130 there in the right way. 2022. I did originally think immobilized enzymes was going to come up in answer to your question. But enzymes, I feel like with the enzyme side of things, enzymes came up so high in paper one that if they're going to do immobilized enzymes, it will be potentially more about where does the source of the enzymes to be immobilized, where could they come from, and looking at batch. That's right, you were meant to reject it. No, not accept it. You're meant to reject the null hypothesis. Do you need to know about K and R strategies? No, you do not, Ted. You do not need to know that. Guys, I am going to finish now. And the reason I'm going to finish now is because I need to make sure my little girls get to bed and I'm on bed duty tonight. So have a good one. I wanted to do this earlier so that I could, one, make sure you guys don't just keep going into the evening. I don't want you to have a late night. I want you to have an early night. I want you to be able to rest. And tomorrow will be what tomorrow will be. It's going to be hard. Be aware that it's going to be hard. It's going to be challenging. It's meant to be challenging, and that's okay. So the key thing for you to note is that when you walk into that exam, take a breath, read the question, do your best that you can do, and just walk out that exam going, right, now on to the third one. Okay? Make sure, please make sure you have a good night's sleep because your brain needs time to tidy all the mess that's inside it right now. So the best thing you can do is one at 8 o'clock, I believe Miss Eastrick is doing one now. It is AQA based, but there will be a little bit of overlap, but it's not specific to your exam. So you could potentially watch a bit of her stuff, but I would say it is geared for AQA more than OCR. So try your best. I will put out a good luck one message for you. And if you are coming out of that exam and feeling that it went hard, then that's okay. That's what's important. I won't be able to see the paper. I won't see it, I don't think, until maybe Tuesday by the time I get to see it, which will be really rough because paper obviously 3 is on Wednesday. So please do let me know how you get on, what questions come up. That's what I'm more interested in, the topic areas that come up, because if they haven't come up in one, they haven't come in two, they'll come up in three. So good luck, everybody, and have a great evening, and make sure you rest up. Don't have a long night. You'll actually do worse if you have a long night. I promise you, you will do worse than if you have a quick cram in the morning before the exam. You're better to have a early night and cram in the morning than you are to do a late night here. I've never seen a C go down to 20%. C can normally go down to about anything from 47 up. It's normally about 53 for a B. 46 maybe at the lowest point. Actually, I should know this. I do have it somewhere, but I cannot think of the top of my head.